Hi, I'm Jonah Comstock, Editor-in-Chief of Harbour Forum. I'm here with Trevor Martin, CEO of Mammoth Biosciences. Hi, Trevor.
Yeah, thanks for meeting with me.
We're, of course, here at JP Morgan 2024. Tell me, first of all, a little bit about yourself and about Mammoth.
Yeah, so Mammoth was founded about 5.5 years ago to deliver on what we see as the true promise of CRISPR and genetic medicines in general. And that's to really be able to create one-time permanent cures for genetic disease and really enable individuals that have these very debilitating diseases to go the rest of their life after a one-time injection with not having that disease.
So this is a big year to be in that field. Obviously, just last month, there were two big FDA clearances for gene therapies. How does how is what you do kind of different or similar to that? And what how does the kind of current excitement and environment around gene therapy affect you guys?
Yeah, it's a very exciting time, and I think it's really, really awesome to see the very first approval of a CRISPR-based therapy in the United States. Obviously, that's a ex vivo therapy, and I think what we're excited about is building on all of that work to really start going towards in vivo therapies, so these kind of one-time permanent cures for genetic disease with a single treatment. I think that what we're excited about there is that we've been innovating in terms of the CRISPR payload itself. So, like new CRISPR systems that go beyond Cas9 that you probably heard of, that is probably the most famous CRISPR system, and really leveraging natural diversity combined with tons of engineering to create new CRISPR systems that, for example, are very, very, very small relative to the original Cas9 systems. And that means we can actually deliver them better for these one time cures.
So what? How far along are you on? How many things do you have in your pipeline? How close are you to getting into trials and things like that?
Yeah, so we're preclinical, and we are really excited that we're starting to generate in vivo data through things like NHP studies with these brand new CRISPR technologies. So these again are just from totally different parts of the tree of life of CRISPR, and this is the first step, of course, to then taking those into the clinic so you can actually address unmet need. And the types of unmet need we're most interested in are things, especially extrahepatic, so things that are going beyond the liver. So like the brain or muscle or heart. And this is where there's a huge unmet need because it's really difficult for Cas9 and these like legacy technologies to actually be delivered there.
And that's a biological reason.
Well, because the systems are actually so large. So the most accessible way of going extrahepatic right now is AAV. And this is a delivery method that has a very strict size limit. So when you're doing ex vivo editing or even doing liver where you can use this other technology called LNP, there's a little bit more flexibility on the size. It's still better to be small, but it's not as hard of a kind of barrier if it just doesn't work like it does with AAV because there's this very strict limit. So for these applications where you want to leverage something like AAV, having these ultra compact systems is what we call them, things like CASV or NanoCas, is a huge advantage because not only does it fit into these AAV vectors, but because it's like a third or less the size of Cas9, it actually has all of this extra space. So you can use that for a bunch of different things. One of them being non-double strand break methods that you might be familiar with. So things like gene writing or epigenetic editing or base editing.
So at a show like this, are you looking for partners, for investors, for other folks who are doing similar things that you can learn from? What are you what kinds of meetings are you having here besides with journalists?
Yeah, of course, that's one of them. I think the great thing about this type of meeting is the diversity of meetings that you can have. So I think we always try and cast a wide net, talking with people in the space broadly, of course, journalists, kind of sharing our story, sharing what we're excited about in the CRISPR space, talking with banks, investors, analysts, and really just making sure that everyone understands the exciting work that's being done in the CRISPR space broadly, and also, of course, at Mammoth in particular, to push the envelope towards what's next in CRISPR.
And speaking of what's next, the other thing that we do at this show is we kind of get our finger on the pulse of what's coming up, what people are excited about. What are some things? I mean, it's early, it's day one. What are some things you're hearing or are excited about hearing more
Yeah,
about?
it's a great question. It is day one, but I think there's cautious optimism in the air. I think people are really excited. Maybe it's just the start of the year. But I think people are really excited about what the year holds because regardless of the macro factors, and I think maybe there's encouraging signs on that end, we'll see. I think that this is an amazing time in biotech. There's just a tremendous amount of innovation, both in CRISPR and at Mammoth, of course, but also more broadly. And so I think people see that there's huge opportunities here, and this isn't a case of like, oh, there's no innovations. Like, where do we, want to focus our time and energy and money? There's tremendous amounts of really exciting work that's being done to meet unmet patient need. And I think that that's something that people are looking forward to over the course of the year.
One of the big outstanding questions when it comes to cell and gene therapy is what's the business model going to look like,
Yeah.
and what are the infrastructure for delivery going to look like, and how can we build that fast enough to keep up with what's coming out? So, what is your take on that from Mammoth's perspective, and how are you thinking about sort of the yeah, both the business model and the delivery infrastructure?
Yeah, I think on the business model side, I mean, the number one thing first is you have to create a functional drug that cures the disease. And I think when you're going after things like we are, where there's just such massive unmet need, I mean, at the end of the day, you're saving the healthcare system tons of money in addition to saving lives, right? So I think from that first principles view, I think it's obvious to us that we'll find ways to make sure that patients can get access to these medicines because it's just the unmet need is so high. I think over time, definitely, I mean, even personally, I want these types of new biologics and new genetic medicines to be as accessible as possible. So I think that's a great goal, but that becomes more of a society level question of like, how much do we want to invest in these so that make sure that there's higher access? As a company, what we can do is actually cure the disease and actually make sure that that's available. And from a personal perspective, though, access is a huge deal.
Yeah, I mean, you never want to lean on if you build it, they will come. But if you do build a certain, I mean, if the folks doing what you're doing build enough of these therapies, then the system is going to figure out how to get them to patients.
Because A, you're helping the patients with massive unmet need, and B, even from a napkin style analysis, you're saving the healthcare system tons of money.
The other big thing that I think we'll be hearing a lot about here is AI and how it's changing R&D. Do you use any AI at Mammoth? How are you thinking about sort of that, and has that changed over the last year?
Yeah, in a previous life, I was a statistician, so very familiar. We've been using AI, machine learning, whatever you want to call it, for over a decade now. That's actually how we find and characterize these new CRISPR technologies. We have these giant metagenomic databases of billions of proteins, and this is from sampling and sequencing all sorts of samples. It could be soil, it could be this table, it could be something more exotic like a jungle and a river somewhere. But all of these different samples contain these microorganisms like bacteria and viruses. And these microorganisms harbor potentially new CRISPR technologies because they're actually the adaptive immunity of many organisms, most famously bacteria. So we have to sort through these giant data sets. So we use our own proprietary machine learning techniques to do that. And then we combine that with massive high throughput experimentation. So our office over in South San Francisco that you might have driven by on the way here has these large robotics facilities where we can actually do high throughput experimentation, combine that with machine learning to actually find and characterize potentially brand new CRISPR technologies like these ultra compact systems like Cas9 and NanoCas.
How have you seen kind of over the last year or so the mindsets in the industry around AI and AI adoption change?
Yeah, I think it's I mean, things always go through some cycles. So I think first it's like, what is this? And then it's this like race to find different applications. And now I think we're getting to the part of the cycle that's most exciting to us because that's what I think matters the most, which is, okay, what does this actually make a difference in, right? Like, you don't want to just like slap AI on different things. Like, what are you actually going to use it for that's going to make a difference, either for building the drug, for patients, for finding the patients, whatever it is, right? Maybe multiple different applications, but what is the AI actually going to enable that it wouldn't be possible without it? And that's, for example, for us for the last decade, leveraging these different machine learning and AI techniques to find and characterize these new CRISPR systems, seeing which ones work and which don't, and then getting better and better at it over time.
Awesome. Any final thoughts? Anything we haven't mentioned yet that you're excited about?
Yeah, no, I think one of the things that I'm really excited about that goes back to the cautious optimism is that, so we have the first US approval of a CRISPR-based therapy. There is immense potential. Everyone, of course, has heard about CRISPR and, many people are excited about it. But I think what's really, really interesting is that we're still, I think, in the very early days, which is so exciting because already we're seeing, immense potential benefit. And I think it's all about realizing that benefit and taking CRISPR to the next level, going extrahepatic, going beyond double strand breaks, and really eventually even going beyond rare disease. And I think that is going to be really transformative. Imagine a world where we cure disease rather than treat it. And that's something that's not just, well, it is kind of sci-fi. It's fun to think about, but it's something that we can actually realize and actually, do in the real world.
Awesome. Well, thank you so much, Trevor. It's been great chatting with you, and good
Yeah,
luck.
thank you.